All right, hello, my name is Kailee Mendiola. I did some research over the summer at Notre Dame through a REU, and one of the things we were looking at were calcium signaling dynamics and lymphatic endothelial cells. So we were looking at the signaling dynamics between these cells as we were looking mainly at these endothelial cells undergo a sort of regeneration. It's called lymphangiogenesis, so it forms new lymphatic vessels from existing vessels, or it's sort of sprouting. And by looking at the specific calcium patterns within the cells, you can determine what kind of characteristics the cells have, whether it is migratory or if it is more of in a proliferative state. And so by observing these patterns in cells under different conditions, we are hoping to actually influence them ourselves. So some of the preliminary work done is both done by collaborators and students that were already in the lab. So our collaborator, Dr. Chang, has this ion exchange membrane that he used to induce calcium signaling in cardiomyocytes. So he used about 45 volts to induce that response. So we wanted to see if we could get a similar response from the LECs, as they are different cells. And one of the things noticed by students in the lab is once plated in a PDMS microfluidic chip, they noticed a correlation between spiking between the cells. Now, while the pattern was irregular, if you notice in the figure 2 how there seems to be random spiking, so the green lines indicate that there was correlation between two different cells spiking. And while it was very irregular, we noticed there were spiking, so we wanted to see if we could pattern it in a certain sort of way. So could we characterize how the signaling moved, and by different methods that are known to affect calcium ion movement? And by characterizing, could we then use what we know to influence their movement in certain ways to then affect their movement in a way wanted? So 3 main things were done that we will go over today, which is a wound assay. We added acetylcholine in different concentrations, and we also depleted the environment of calcium ions. So first, what we looked at was a wound assay where we scratched a cell monolayer of lymphatic endothelial cells with a pipette tip. This was started off as a migration assay, but the calbrite ended up being cytotoxic in the long term, as there's a mechanism that I know, but not off the top of my head, that ends up killing the cells long term, but it lasts for about 30 minutes to a couple of hours, depending on the concentrations. And so the cells that came into direct contact with the tip when it was scratched died, as you could see in the picture on figure 4, how the calcium was very intense. And as you know, calcium, or as you may not know, calcium is also an indicator of cell death whenever it is just brightly lit like that. But one of the things we noticed in figure 3 was that the correlation ended up following the pattern of the wound. It when accounting for lag, it would follow in a sequential order. Of course, the code can still be optimized for use. As you can see, the program is telling us there's communications between the 2 different sides of the wounds, so that would need further investigation on how to optimize the code better. Next, this was supposed to say acetylcholine. I'm so sorry, but we added acetylcholine in 4 different concentrations and incubated them there for about 24 hours, I believe. And why we used acetylcholine is that it's a neurotransmitter that binds to intracellular pathways that open up communications, because this encourages dilation of different vasculatures, such as blood vessels, lymphatic vessels, and other vessels in the body. And the 4 different concentrations we used were 3, 20, 50, and 100 molar. And while there may not have been a significant increase in the number of cells speaking, in the fraction of cells spiking, there was definitely an increase in the correlation. So one of the main portions of the project, this part took the longest because we believed this was the most important part of our project, was ion depletion. So IEM technology is where we have this current running through 2 different electrodes. And one of the negative electrode, I believe, has this membrane that is selective only to calcium ions, so that the calcium ions can flow into as close as they can to the electrode and kind of get stuck there. So it's kind of depleting the environment slowly but surely as it goes away. So one thing we noticed between the 2 differences is up here, before the voltage was introduced, the cells were spiking, similar to how the preliminary findings found, how it was just irregular. But once it spiked over here, of course, majority of the cells in the bottom got washed away, but up here where there are still cells, it is very brightly intense green, meaning that is very high intense correlation, internal spiking and correlation. But since we also used 45 volts, and 45 volts is typically for cardiomyocytes, they ended up intensely spiking, then dying. So of course we would want to tweak the voltage to be better optimized for the lymphatic cells, because while cardiomyocytes are muscle cells, they're more familiar with the voltages. Lymphatic cells don't typically experience a voltage in the body, well, like the cardiomyocytes do. What we did notice is there was a significant increase in correlation. It wasn't just more cells started spiking, it was the cells started talking at the same time. And by getting them to speak at the same time, that really shows that they might actually be talking to each other rather than just spiking and being proliferative by themselves, and instead communicating to other cells that they're also experiencing the same conditions as well. So what we noticed is, while under homeostasis, it was irregular. They were just spiking kind of like twinkling lights. While we started producing these different effects and methods on the cells, they started reacting in different ways, such as the sequential linear as the wound assay was applied, the acetylcholine as spiking was increased, and increased spiking and correlation of it. And as ion depletion was introduced, simultaneous wave-like calcium signaling. So depending on what functions we want to use later, we can then introduce these different methods to then get the spiking patterns we would want, rather than just seeing what happens. And depending on how much of a voltage or how much acetylcholine was introduced, that also gave us a proportional response, as that's why in acetylcholine we compared the 3 micromolars to the 100 micromolars. And that's why we also want to observe different voltages within the cells in the ion depletion movement. Next would be to observe effects of flow. By the time I was leaving, they were getting ready to set up flow. I did not have the pleasure of being able to observe it, but I assume it went well. I also would have run experiments on the other endothelial cells, such as blood endothelial cells and endothelial progenitor cells, such as endothelial colony forming cells, as we would want to see if this is a similar behavior between all endothelial cells or just lymphatics, to understand how we would need to tweak our approach anytime we change a cell type. And thank you to Dr. Chang and Dr. Zartman, as when I was there, they gave wonderful suggestions and wonderful guidance. to my advisor at UTSA, as he is the one that connected me to this lab here at Notre Dame, and everyone that gave funding, and especially the paper trail, as I really think the mission for the paper trail symposium is really cool. Yeah.
Thank you so much, Kailee, for presenting your research with us today, and we're happy to have you. Do we have any questions in the chat? Anybody online who would like to ask? Looks like Chris was first. Raise your hand if you want to unmute and ask your question.
Hello, thank you for the great presentation. I know you mentioned a possibility of doing different cell types separately to see if they have the same response. Do you think there'd be any interest in looking into cell types together in co-culture? The lymphatic system, of course, works very closely with our blood vessel system, and they likely do have some crosstalk. Would you be interested in looking at how those two systems might co-communicate in this manner?
So I also know that there's smooth muscle cells within the lymphatics as well. So blood endothelial cells and lymphatic endothelial cells is one thing to look at, but I also think probably the first step before that would be seeing how they interact with the smooth muscle cells belonging to the lymphatic vasculature.
Perfect. Additional questions? While we wait to see if there are any other questions, you want to talk about what your interests are and what you would think about studying or researching your next steps?
So I really enjoy the bioelectricity aspect, or cellular communications as well. So back in my research lab here, I also investigate the effects of electrokinetics within different biological systems. And so I really enjoy that. And I plan to pursue my PhD by going to grad school.